All right. Hi, everyone. I think it's good as time as any to get started. So thank you all for coming and joining us for this event tonight. I'm super excited about it. I guess I'll just give you a little intro to me, and then I'll give you an overview of what this event will look like. So my name is Megan Hochstrasser. I'm the Education Program Manager at the Innovative Genomics Institute. And if you're not familiar with the IGI, we're a partnership between UC Berkeley and UC San Francisco. We were founded by Jennifer Doudna, who's known for developing CRISPR technology, with the idea of making sure that CRISPR gets out of the lab and into the real world to solve big problems and really help people. So part of our work is actually on sickle cell disease, and we also do a lot of outreach and engagement surrounding CRISPR. So tonight, I think, is a natural union of our different activities. Okay, so the general format will be me talking for the first half of the event or so, and giving you a little bit of background on CRISPR and sickle cell disease. And then I'll play a short virtual reality tour that will hopefully kind of bring some of these different ideas together into one place to connect the dots. And then after that, we're going to have a really exciting panel discussion with a bunch of really amazing guests that we have for you tonight about the ethics of treating sickle cell disease and the racism that the sickle cell community has actually faced over the years, and what we can do about that. So I think without further ado, I'm going to get started. So everything tonight really begins with DNA. DNA is the instruction manual that all of our cells have that helps them function and makes the body work. And it controls traits or characteristics about living things. And I'm showing you here just a few examples of traits that you can learn about in 23andMe, which you probably already know if you have these traits or not, but by doing 23andMe, you can see how they're actually associated with your DNA. And this ranges from everything from like whether you have dimples to whether you're afraid of heights. And these are kind of fun and like frivolous little things to learn about in your DNA, but there can also be mutations or variants in your DNA that cause harm. So I just took a few more of the disorders that 23andMe actually tells you about. So some of these are more known, like cystic fibrosis. I think that's a pretty well-known genetic disorder, and others are much more rare and people don't know them as much. So there are actually thousands of different diseases that are caused by mutations in your genome. And the one that we'll be talking about tonight, of course, is sickle cell disease. So this is a condition caused by a mutation in DNA that makes the red blood cells turn into this sort of half moon or sickled shape. And because of this shape, cells don't really flow very well through blood vessels and they get stuck. So they kind of pile up on each other, and the cells don't live as long as a healthy red blood cell should. And so people who have sickle cell experience these really awful pain crises. They become anemic, and there can be tissue and organ damage when oxygen is not reaching the tissues where it should be getting. So this condition affects about 100,000 people in the US and actually millions worldwide. And it's predominantly something that impacts African Americans or people of African ancestry worldwide. And that actually really plays into why there has been kind of chronic underfunding of sickle cell research and poor care and management of pain for sickle cell patients historically, which again, we're going to get into in the second half of the event. So last, the only current curative treatment for sickle cell is a bone marrow transplant. And this, while it can actually and has cured people of sickle cell disease, it's not done very often because it's a kind of grueling procedure and you really need a close match. So the donor will often be like a sibling, and that's not always doable. So it's kind of a tricky procedure and not done very often. So I think the patients are really in need of other options. So just to go into kind of the nitty gritty of what causes sickle cell disease, it's actually caused by just one letter in the DNA being mutated. So you'll see here, there's just this one little variant in the entire genome that can cause this condition. And this one little switch actually has to be on both chromosomes. So the chromosome from the person's mother or their father both have to have that mutation in order for someone to have the disease. And the reason that this typo in the instructions actually causes problems is because of this protein called hemoglobin. So hemoglobin is responsible for carrying oxygen, picking it up in the lungs and dropping it off later in the bloodstream. So this hemoglobin molecule, just for reference, I guess it's made of a couple different parts. So it has subunits, and the that causes sickle cell is actually in the beta subunit. But for ease and simplicity, I'm just going to talk about the hemoglobin protein and the hemoglobin gene, just in general. And so in a healthy person who doesn't have sickle cell, their hemoglobin is these little freestanding molecules that float around happily. And then in sickle cell, the hemoglobin actually gets sticky. So that one little mutation that happens in the DNA causes a little mutation in the protein that makes it stick to itself and make these big fibers. And that's what distorts the cell shape. So because this is a genetic disease and it's caused by a mutation in DNA, we can, in theory, cure the disease if we can fix that mutation in the DNA. It's not that simple, but hopefully we'll learn a little bit more about how this could work. So that's what brings us to CRISPR. So CRISPR is a system that lets us rewrite DNA inside living cells and organisms like humans. And it works. We'll get to know a little bit more about this in the VR tour, but in general, we use a CRISPR protein. That's what I'm showing you in green here, that we can program to go anywhere in the DNA that we want. It'll search through all the DNA and then find the target. So we can target it at the sickle mutation, and then it'll make a cut, and that triggers a change in the DNA sequence. So we're actually able to replace that kind of broken copy of hemoglobin with a healthy one. And just to give a little more of a kind of a practical explanation of how this could work, what would physically happen is oh, sorry, that's the next slide. This is another kind of metaphor. If it's easier to think about CRISPR as kind of like a text editing tool, you could think about this sentence here, like say that's a DNA sequence. You could have a mutation in the DNA. So here it shouldn't say twinkle, twinkle, battle star, right? Unless you're a sci-fi fan. So we could use CRISPR to change this sequence into the correct ones, twinkle, twinkle, little star. So that's why people kind of compare CRISPR to like a text editor for the cell. Another metaphor that I think can also work is this genetic surgery idea. Like we can cut DNA, we can replace DNA. I think that can work too. So whatever helps you understand it better, I think go with that metaphor. And so next, this is the slide about kind of how this would practically work. So right now, say this is our patient. Our first step to using CRISPR as a therapy for this patient would be to take cells out of the person. So we would remove cells, and in this case, we need the blood stem cells because those are the cells that are going to make all of their new blood cells over the course of their life. And we take them out and we can put them in a lab dish and keep them in the lab. And if we looked at the DNA in these cells, we would see that they carry this sickle cell mutation. So this one letter is switched. So the next step would be to edit those cells. So we would add in CRISPR enzymes, that little protein that I showed you before. And once that's in the cell, we could check these cells and see if our editing was successful. So if we looked at their DNA, we would hopefully see that that mutation had been undone. So we would have the healthy sequence instead. So once we're confident that our editing has worked, we would return all of the cells to the patient so that they can hopefully engraft, meaning they would kind of hang on and stay in the patient and make all kinds of new healthy red blood cells for them. So that's kind of a general overview of the process. I think it's important to note that this is not really the ideal process. Ideally, we would just be giving someone like a shot that would be curing them of a disease, and we wouldn't have to do this complicated procedure of removing their cells. But right now, this is really what's possible. And because this is possible, this is why sickle cell is kind of one of the first conditions that is being tackled using CRISPR because we can reach these blood stem cells. So as much as there, improvements could be made to this, it's actually already helping people. So this woman who I'm showing you is named Victoria Gray, and she has become kind of an amazing hero in the CRISPR world because she is the first person in the US to receive a CRISPR treatment for sickle cell. And NPR has been following her story, which I recommend you check out their coverage. They even have audio interviews where you can hear her speak directly. And it's been amazing to see this. So she received this CRISPR treatment, I think a year or two ago now, and she had very severe symptoms before this, but now it seems that she has not had pain crises and doesn't need transfusions. It seems like she seems to be mostly cured of the disease. And I'm very hesitant to say cured because we really have to follow people long term and make sure that this is a permanent change and that there aren't side effects that we haven't been able to detect yet. But it does seem extremely promising. So I think everyone is very heartened to see this data. And I'll also note that this is not someone that we're treating. There are a variety of groups of scientists around the world who are all kind of simultaneously looking into different strategies for treating sickle cell. And they're all a little bit different, but I think that having these many different irons in the fire is ultimately going to benefit the sickle cell community. So hopefully we find methods that are really optimal. Okay. So that's kind of a brief overview of both sickle cell and CRISPR. And I hope that this virtual reality tour that I'm about to show you can kind of bring some of these concepts together for you. And I'll just note that this is really something that's designed to be immersive, where you're wearing a headset like this person in the picture. And hopefully if we're able to do in-person events soon, you guys can all come to some of our events and try this yourselves, because it's a very different experience to control it. But I think the video will also give you a good impression. So with that, I will switch over to the tour, and this part is pre-recorded to make sure the timing works. And I will get it started. Our simulation begins with a theoretical sickle cell patient. On the left is our patient, and on the right is the patient's chart. This shows us how they're doing. We can see that they have a hemoglobin mutation, sickled cells, low blood oxygen, and they're in a lot of pain. Looks like they're ready for our treatment. Let's dive in and get a closer look at what's going on. Here we've landed on a bone. There are blood vessels going in and out of it. Since sickle cell disease affects blood cells, why don't we check inside this vessel to see what's happening? Alright, now we're inside the blood vessel. You can see on the left, before this vessel splits, there's pretty good blood flow. But on the right, all of these cells are getting stuck and piling up on each other. It's pretty clear here that a lot of the cells are misshapen. There are some rounded, healthy red blood cells flowing through, but many of them are distorted and shaped like half moons or sickles. This is where sickle cell disease gets its name. Because of their shape, these cells get caught and block blood flow. This cuts off oxygen to the downstream tissue. That lack of oxygen is what causes the intense pain that this patient is experiencing. Let's go inside of one of these cells to figure out why it's sickle-shaped. Alright, we've landed inside the sickle cell. You can see that we're surrounded by a lot of copies of this one particular protein called hemoglobin. Hemoglobin's job is to carry oxygen inside red blood cells. In this case, it doesn't seem to be working the way it should. It's sticking together in these long fibers. These stiff fibers of hemoglobin build up and distort the cell into its sickled shape. You may have noticed that these hemoglobin proteins have a little red dot on them. This represents the sickle mutation. When we see a mutation in a protein, it means there's a typo somewhere in the instructions for that protein. The instructions for a protein are called a gene. In this case, there's a typo in the hemoglobin gene. Since we want to repair the gene, we'll need to go find the DNA. So let's zoom out and see if we can help this patient. Okay, we're back on the surface of the bone, but we need to find the cells that give rise to new blood cells, and these live inside the bone, in the marrow. In the bone marrow, you'll see a lot of red blood cells and these very special blood stem cells. The blood stem cells are these whitish purple cells. They're the cells that will divide over and over again to make new red blood cells. If we can repair the sickle mutation in the stem cells, then all of the blood cells that come from them should be healthy. Let's dive into one of these stem cells and see what we find. Okay, this is the inside of the stem cell, and there's a whole lot going on in here. But what we really care about is the DNA, and the DNA is stored in something called the nucleus. That's this big ball in front of us that we need to zoom into in order to find the genome. All right, here we are. We've found all the DNA, but we're just looking for one gene, the hemoglobin gene. The hemoglobin gene is actually this sky blue colored stretch of DNA right in front of us. And you'll notice this little orange spot on the DNA. That's the mutation that causes sickle cell. Now we've designed a CRISPR enzyme that's already been added to the cell, ready to edit the sickle mutation. We've also designed a piece of DNA that contains the healthy version of the hemoglobin gene. We're going to get the cell to paste in this donor sequence to replace the mutated copy. Now let's trigger the CRISPR protein and watch genome editing in action. The CRISPR protein finds its target, unwinds the DNA, and makes a cut right next to that mutation. The cut activates the cell's repair system. One of the cell's repair proteins will bump the CRISPR protein out of the way, grab that healthy sequence, and copy it over the original mutated gene. All right, so we've done it. We've replaced the sickle mutation with a healthy sequence. I know that was quick, so let's watch it one more time. I'll just hit the replay button. The CRISPR protein finds its target and makes a cut all the way through both strands of the DNA. The repair system displaces CRISPR and pastes in that healthy DNA that we provided. And I'm not showing you all the details of the DNA repair process because it's a little bit complicated. But in reality, we do provide a piece of donor DNA for the cell to use as a template so it can insert the healthy sequence. So it seems like we've fixed the DNA in this cell. And in real life, we'd have to do this treatment to a lot of cells at once, not just this one. So let's go see what would happen if we were able to make this DNA edit in a lot of the stem cells. So if we zoom out to the bone marrow, you'll notice that already a lot of the cells look pretty healthy. I don't see any more of the sickled cells that were here before. But the big problem was happening in the vessel, so let's go check that out. All right, it looks like the blood is flowing smoothly. I don't see any more sickled cells. That blockage is gone, so oxygen is probably getting to all the tissues, and hopefully this person is no longer in pain. Let's go all the way back out to the beginning and check our patient's chart. Great, the DNA is edited, the cells are no longer sickle-shaped, oxygen is flowing, and the patient is no longer suffering. This is a type of treatment we hope to offer someday. At the Innovative Genomics Institute, we're working with collaborators across the University of California system to make this a reality, but this treatment is not yet available. If and when our CRISPR therapy proves to be both safe and effective, we hope it will serve as a life-changing option for the sickle cell community. Okay. Thanks for watching. I see some questions in the Q&A that I think I can tackle. I think they were a little bit addressed in the video, but I can go through them quickly here. So the first one of the questions is, how are the stem cells actually collected? And these stem cells, we don't have to go all the way to the bone marrow to get them. So you can do something called mobilization, where you can give patients mobilizing drugs that makes their stem cells come out of the bone marrow and into their bloodstream. And then you can just collect the cells directly from their blood, which makes it a lot easier. So that's how we do that. And then the other questions are, um, let's see, are you only repairing some of the DNA and some of the cells? And are you only repairing blood cells? So we actually want to repair the DNA in as many of the stem cells as possible, because those cells are going to divide and divide and make new red blood cells. So if we can edit that DNA in the source cell, all of its children cells basically will have that corrected hemoglobin. So we're trying to get as many as possible, and we want it in the stem cells. And we are only trying to edit blood cells. So if we edit these progenitors of the blood cells, we're just impacting blood. Okay. I think that's a couple of the questions. Let me just see if there are a few more. Okay, and then I will answer two more here. So how quickly does the edit show improvement? So the editing process can take like maybe a day or two for the DNA to actually change. And then I think it would take several weeks probably. I don't know exactly, because again, this has not really been done very often in humans yet. But I think over the course of several weeks to months, that's when you would see the older sickled cells kind of disappearing, because they don't live that long anyway. They would kind of go away. And hopefully, as these stem cells divide, they're making new healthy red blood cells. So it would take a little bit of time to see any clinical or symptom level impact, but less than a year, I would say. And then the other question, which is a really good question that I didn't really mention, is why are people of African descent, why do they have a greater chance of getting this disease? And the answer to that is because it's kind of an interesting thing that is often taught in biology class, as an example of a genetic disease that actually came about because it had an evolutionary advantage in some ways. So in Africa, malaria is a big problem. And for whatever reason, I don't think it's actually known very well. If you have just one copy of this sickle mutation instead of the two that will give you the full disease, you don't have symptoms, but your cells are actually protected from malaria to some extent. So it's known to be like kind of beneficial to have this one copy. And so that mutation is able to persist in the population. But then unfortunately, if you have two copies, like both parents carry this, you can get the disease. So that's actually why it's associated with people of African descent. Okay, so I think I'm going to stop with science question and answer for now, and just note that this virtual reality experience was made with these two really great collaborators. Dynamoid is a company in Oakland, and the Lawrence Hall of Science is our local science center in Berkeley. And if they're open again, maybe in the summer, this is actually going to be part of a planetarium show that you can go see. And it's a little bit more immersive to see it projected above you than to see it on a 2D screen. So I recommend doing that. And then for those of you who might happen to have an Oculus Rift or HTC Vive VR system, this is available on Steam. So it's part of this program called 10K, like 10K. So if you actually have those systems, you can download this. Okay, and then one note before we get to the panel that I just wanted to make very clear is that everything I'm talking about, like what we're developing at the IGI, what Victoria Gray, the treatment that she underwent, that is all this one kind of genome editing called somatic editing. And that is distinct from another approach called germline editing. So the key differences here are that with somatic editing, the changes that we make are not going to be passed down to the next generation. So if we edit Victoria Gray and she has a child, her child could still have sickle cell. So she could still pass that on, because we're only affecting the most of the cells in her body, not her germline cells, which are like sperm and eggs. So so it's only she's affected. But with germline editing, if we were to edit her eggs or an embryo, that would be passed down to the next generation and all of the subsequent children and generations. So that's like a permanent change in someone's lineage versus a change that just affects one individual. And this comes up a lot in discussions about CRISPR and ethics because one is a really long lasting impact, one is not. And then in one case, in germline editing, when you're editing an embryo, it's pretty well established that CRISPR does not work very well in embryos, or not many labs are working on it, honestly. So I think it doesn't work very well yet. So it's not thought to be safe. And so most people are either morally or scientifically opposed to even trying CRISPR in embryos at the moment. So yeah, I just want to make sure that that was clear in case it comes up. So I think with that, I will pass it on to our awesome moderator and introduce him. So this is Kevin Dockson, and you can turn your video on. Hey, Kevin. Kevin Dockson is the science communications specialist at the IGI. We both got our PhDs in Jennifer Doudna's lab studying CRISPR, so I've known him for a while now. He's very thoughtful and has a lot to say about ethics and how CRISPR should be used. So I think he'll be an awesome moderator for our panel. Take it away, Kevin.
Great. Well, thanks, Megan. Thanks for an exciting look at how researchers are preparing to use CRISPR genome editing technology to address sickle cell. And now that we have the background scientific knowledge about what's happening on the molecular level, let's zoom out and look at the societal level. So we're fortunate to be joined by a panel of experts to discuss a wide range of topics today, including health disparities in the US, race and racism in medicine, and access to emerging technologies. So I'd first like to introduce Marilyn Bafo-Bani, a second-year doctoral student in the Department of Sociology at Rutgers University. Her research interests include health disparities, race, ethnicity, and immigration, and the ethical, legal, and social implications of genetic and genomic research. Marilyn holds a Master of Bioethics from the Perelman School of Medicine at the University of Pennsylvania and a Bachelor of Science in Psychology with minors in Health Studies and Peace, Justice, and Human Rights from Haverford College. Next, I'd like to introduce Cassandra Tremnell, a patient advocate, speaker, consultant, and the founder and executive director of Sickle Cell 101. In 2014, Cassandra received her Sickle Cell Educator Certificate issued by the Department of Public Health. She is responsible for developing and creating the educational material and curricula presented by Sickle Cell 101, a San Francisco area-based nonprofit organization that specializes in sickle cell education, geared towards multiple key community stakeholders, including patients and healthcare providers. Educational material covers topics pertaining to sickle cell disease and sickle cell trait and is disseminated primarily through social media. Cassandra is a board member for the Global Alliance of Sickle Cell Disease Organizations and sits on committees within the NIH, the HHS, and other entities that have a stake within sickle cell. She received a BA in Global Studies, Social, Political, and Economic Development from Sonoma State University. And Cassandra says that her passion for sickle cell education and advocacy stems directly from her living with sickle cell. I'd next like to introduce Dr. Monica Filvia Moher. Monica applies a cultural lens to storytelling and science communication to engage underserved communities with science, especially to Puerto Ricans and Latinxs. As a science communicator, she draws on her training, a PhD in neurobiology, a personal background and culture, a woman from rural Puerto Rico, to make science relevant and relatable to marginalized populations. She works with the nonprofits Ciencia Puerto Rico and iBiology, where her efforts include serving as editor-in-chief for the publication of more than 400 scientist-written popular science articles and op-eds in major newspapers, co-editing the first collection of culturally relevant popular essays and stories about science in Puerto Rico, producing a series of short films changing the narrative about scientists of color, and training hundreds of scientists in culturally relevant science communication, among others. She obtained her BS in human biology at the University of Puerto Rico in Bayamon and her PhD in neurobiology at Harvard University. So I'm really excited to talk with all of these fantastic women about such an important issue today. And Megan needs no introduction. Thanks, Megan, for really getting us all on the same page before this discussion. And the first question I'd like to start off with is for Cassandra. So could you describe what it is like to live with sickle cell disease?
Hi, Kevin. Thanks so much for having me here today. Absolutely. I'd love to share. Um, so I think there are two main symptoms that really kind of just affect me on a day-to-day basis, and they are pain and fatigue. So fatigue kind of feels like everything is just the most simplest tasks are very hard to do. So like picking up a pencil or getting up to grab some water. It feels like you have kind of like just this um, 50-pound sandbag on all your limbs and, just your whole body. And it's just like you feel sluggish and, just like the simplest task, like I said, it's very, very difficult and you're tired all the time. So that's what fatigue feels like to me. And that's something I deal with on a day-to-day basis. I'm already tired for tomorrow. And that's just kind of how that works. Pain is actually the hallmark of sickle cell disease. It's what most of us talk about the most because it's traumatic. It can be traumatic. Just to give an example, like today I'm in pain. I have actually a pain crisis in my shoulders at the moment. And you never quite realize how essential your shoulders are, or whatever part of your body that's hurting, until you realize that every time you use them it causes pain. So like, for example, I have a 6-year-old daughter. Picking her up, it hurts. Grabbing a cup, like from a cupboard up high, it hurts. Doing that hurts. Lifting up my purse onto my shoulder hurts. So it just, these small tasks, when it's a combination. Fatigue and pain make even the smallest tasks seem like impossible to do. um and so what I'd consider the pain that I'm in today would be kind of a mild pain, but severe pain is what is I mentioned the word traumatic. That's what's extremely traumatic. And I just to provide context to that pain, because I think pain with sickle cell disease gets understated. Um, People throw around the word pain and think of headache, or but just to provide, I guess, context. So like I said, I have a 6-year-old, and I think everybody knows, without having a child or without undergoing child labor, that child labor is the most painful experience ever. And so being a mother and having gone through it, I can contextualize it and compare the two, and compare, what humankind says is, just the most severe pain ever, and compare it with my sickle cell pain. And just to provide context, just that severe pain is a lot worse than child labor. Child labor, you get to kind of take breaks, you get to recoup, you get a cute baby at the end. Whereas sickle cell pain is just intense pain without any time to recoup. It's the type of pain where you can't sit still. Your body just can't even sit still. You have to keep moving all the time just to try and find comfort, and you don't find comfort. So it's really something. It's really traumatic. And so that's kind of what, and that isn't day-to-day pain for me. Luckily, I don't have severe day-to-day pain. But it is part of the disease, and I still remember it from, when I have it. So that's kind of just to provide context on, just the levels of pain you can have with sickle cell disease. So I hope that answers your question a little.
Great, thank you so much. And that segues nicely into the next question, which, so there's a lot of excitement right now with this technology and the potential it might have for the sickle cell community, but we can't just look at the present, right? We have to consider the history and how we got here. And so maybe this question is both for Marilyn and Monica. Could you give a brief overview of the historical relationship between science and medicine and the sickle cell community?
Thank you again for inviting me here. I would be happy to talk briefly about the history. So the relationship between the scientific community and the sickle cell community is one that, has been described as long and tenuous. The difficulties between the two communities is one that has been difficult, not just only in research settings, but also in medical settings. one consistent complaint that some members of the community raise is mistreatment within the healthcare system, especially in interactions with practitioners. So some say that, you know, they experience lengthy wait times in the emergency department. They're being treated like drug addicts. They're being called frequent flyers, and, you know, their pain is often minimized or not taken seriously. So if they are admitted to the hospital, they often receive the same type of like slow and negligent treatment. And, you know, this poor medical treatment can be attributed to medical discrimination, which is just a facet of other larger issues of racism that are endemic in this country. So, for example, while the cause of sickle cell has been known for over 6 decades, the pace of developing therapeutic and curative options for the disease has been slow moving, especially when you compare it to other diseases that do not predominantly impact people of African descent. This can partially be attributed to the historical lack of research funding allocated to sickle cell, as well as a persistent lack of equitable funding for research, which has continued years and years, even though, you know, various administrations will spotlight the disease and, you know, talk about how it's faced neglect. And if you really look at the overall history of sickle cell, you see that it goes through these periods of waxing and waning celeb status where the disease is used to promote different agendas without these stakeholders really making a concrete, tangible investment into longitudinally improving lives. So, for example, in the 70s, the Reagan administration took up sickle cell disease. And then again, different stakeholders took it up when they were discussing stem cells in the early 2000s. And now again, you know, it's getting a spotlight in this post-genomic era. But I would like to say that while the relationship with the scientific community and the sickle cell community has been historically fraught, it's really important to note that a blanket statement of the sickle cell disease community mistrusts medicine is not really accurate. You know, there is this whole larger network of the sickle cell community that includes people living with the illness, their families, community-based organizations like Sickle Cell 101, for example, and some other researchers and practitioners who are really coming together to try to improve the lived experience. And as, you know, time goes on, more and more stakeholders are coming together to work with this larger community. In the last year, for example, there have been 2 drugs approved by the FDA to treat sickle cell, and clinical trials are underway to find safer therapeutic and curative options. So overall, I do just want to highlight that there's certainly hope that the relationship between the 2 communities can continue to improve, but it's definitely important that people living with sickle cell are present at all tables where debates about their lives are happening.
Great, thank you. And Monica, I think I have a segue question from that, because you've recently written a fantastic paper on this particular topic. And so my question for you is, genome editing for sickle cell is an example of an emerging technology that's impacting a specific marginalized community. And so how does one engage and partner with marginalized groups to explore and address the multiple complex scientific, ethical, and legal issues related to gene editing? You know, how does one avoid perpetuating disparities?
Yeah, first I want to start by thanking you for having me. It's really a pleasure to be here. Before answering your question, I did want to add that what Marilyn described is reflective of larger issues in science and medicine where systemic racism leads to health disparities for Black people and for other communities of color. And so we not only see the issues that she described with patients with sickle cell disease, but this is really a systemic issue. And you know, she alluded a little bit to the need of essentially centering these marginalized communities that are affected by a variety of diseases, in this case, sickle cell disease. And, you know, the first thing that's really important is that we have awareness. We have awareness of the historical injustices and inequalities that exist in science and medicine and that certain communities have faced. We have to also, as, you know, physicians and educators and science communicators and scientists, we have to become aware of our own biases and stereotypes. We're human beings and we all have them. And those biases and stereotypes play into, what are we researching? How are we approaching the problems that we're approaching? How are we partnering or not partnering with certain communities? So working in that issue of awareness is really, really important so that we can center the experiences of these communities. The other thing is connecting with those communities, really listening in, some as a Puerto Rican woman, Latinx woman who passes as white, I can't pretend to understand the experience of not just Cassandra's experience living with sickle cell disease, because I don't have sickle cell disease, but I can't fully understand what it is to live in her skin, to be a Black woman. I can't understand that, but I can empathize with that. I can listen to her experiences and then think about, from my privilege, what is it that I do if I want to partner with people like her, if I want to partner with communities like her, how do I leverage my privilege, the privileges that I have? And so how do we establish those authentic partnerships where, for example, researchers are not just parachuting into a community and saying, I'm the expert, I know the science, so this is what we're going to do. There needs to be true partnerships because, nobody knows what it's like to live with sickle cell disease better than the people who live with it. And so that co-creation, those meaningful partnerships are really important. The other thing is messengers, especially if we're thinking about education and improving relationships between science and medicine and sickle cell communities through education, is like, who's the messenger? Again, somebody like me might not be the appropriate messenger. Somebody like Cassandra might be the right messenger. So that's important. And then the last thing is funding. I think, of course, a lot about communication and outreach and doing that type of public engagement. But if we want to do that right, we need to fund these initiatives, just like we need to fund research in sickle cell disease. We need to fund research in how to communicate about sickle cell disease and gene editing more effectively. And so those are the things that I would say.
Great, thank you. And I know people are submitting questions both on Zoom and YouTube, so keep submitting the questions. We'll get to those in a second. But right before that, maybe my final question is one of access. So, oftentimes you can have academics or companies create a drug and say, here it is, and set whatever price they want. And what do you think needs to be done in parallel right now as the science is trying to become more safe and effective? What needs to be done either in engaging with the community or trying to find economic models that work? What needs to be done to ensure that the people who could most benefit from it receive it?
And you're asking me?
Anyone, yeah.
Anyone, okay. Since I admitted myself, I'll get started. I mean, I think, again, it's about these partnerships. The sickle cell community needs to be part of every single step of the process, from the research, the people who are actually doing the research in sickle cell and CRISPR, to how do we distribute that, how do we test it? Like, these communities need to be part of every single part of the process. Unfortunately, I think when it comes to equity and inclusion, it's often an afterthought. We're doing something, I think, especially in science, sometimes we'd like to think that we are objective. We're not, because again, we're human. But we'd like to think like what matters is the science, but science is a fundamentally human endeavor. And so who participates of the process is important. And if we truly want to center marginalized communities in general, but the sickle cell community specifically, they need to be part of every single step of the process.
Can I just echo what Monica just wonderfully articulated? I actually have that written down as my notes: include patients, every step of the way. And then just to add on to that, education. And education, educating the patient population, and everybody within it. I always say you could have the perfect cure, just an exact replication of exactly what a patient wanted. But if you don't deliver this information to patients and meet them where they are, it might as well be like it doesn't exist. So, and I think, just like Monica said, including patients every step of the way is how you do that. You educate them, and you make sure that they're at every decision making process. And we know what we need. We're the experts on being patient, so we know how to solve a lot of our problems. So just simply having us at the table already resolves a lot.
Great. And so we received a question from Michaela, and maybe this is for Megan. She asked, what roadblocks or challenges are being faced in the process of really making CRISPR a sickle cell cure? So are there maybe technological hurdles that people are really focusing on right now?
Yeah, sure. Let's see. Yeah, I think I would say that the big hurdle for developing CRISPR therapies that will scale and be more affordable is delivery. So this is something that if you hear people talk about CRISPR in general, we talk about time and time again, because it's actually a barrier to treating a lot of different conditions, not just sickle cell. But with sickle cell, as I described before, when you're taking these blood cells out of someone, manipulating them in the lab and returning them, that inherently is expensive. You need people with expertise to handle those cells. It's just kind of a complicated process. So if we were able to deliver CRISPR directly into the body instead of having to do this kind of workaround, that would really be a big improvement. So that's something that a lot of scientists are working on right now. And it's a tough challenge, but I think I've been kind of heartened to see that the NIH and Gates Foundation have just committed to funding, I forget how many tens of millions of dollars, but it's a lot of money towards trying to improve delivery of CRISPR and other, advancing other approaches to both sickle cell and HIV. So I think hopefully we see some big changes on that front in the near term. But I think, yeah, I'm trying to think. There are a lot of different strategies being pursued right now. So I think the biggest hurdle that I can think of really is delivery.
And then one person asked, which year in the future will CRISPR be more developed so that it's available for the public? So I want an exact year, Megan. What year?
Oh, yeah. That's something that's, I really want to make sure is very clear, is that none of this is really available publicly yet, and it probably won't be for several years at least. So the approaches that are in clinical trials now will take a couple years of being tested in just a very small number of people. And, maybe you're learning about clinical trials because of the COVID crisis right now a little bit, but you have to go through several stages where you scale up and you make sure things are really safe. And then at the end of that process, if things are safe and they actually work, the FDA will have to approve this as a real drug or a real treatment. So that could be like five years, 10 years. It can take a while, and it's hard to predict. So I can't say a specific year, but I think sickle cell, I'm hoping at least, will be one of the first therapies that should be available if it continues to go well like it is right now.
And Marilyn, someone asked, does sickle cell disease affect how long you live?
Yeah, so sickle cell disease, Megan spoke earlier about some of these painful crises, as well did Cassandra, but some of the other, clinical manifestations of having sickle cell are the organ damage. And because of, these repeated blockages in cells, which happen in organs, they can really reduce not only just the quality of life, but your lifespan. So people with sickle cell disease are seeing, 20 to 30 years of life lost in comparison to people who do not live with sickle cell disease.
Great, thank you. And multiple people have asked, and I open this up to the whole panel, will CRISPR be used or can it be used to treat other diseases? Either maybe right now in mouse models, or is anything approaching clinical trials? Have any other clinical trials started?
I could tackle that one. Yes, the answer is yes. So there are a couple of clinical trials right now using CRISPR. I would say the one with the disease with the most clinical trials going towards it right now with CRISPR is cancer. So that's kind of a different use of CRISPR. You're not trying to change an underlying mutation that's causing the disease like you would for sickle. But in cancer, you're reprogramming a patient's immune system to try to tackle cancer and find tumors. So that's one. There's also a handful of others. So there's a genetic form of blindness that's being, a cure for that is being tested right now. Trying to think, there are a handful of others so far, including sickle cell. So in theory, CRISPR, because it changes DNA, could be used to treat any genetic condition. It's just kind of a matter of having delivery reagents, like I said, like having a way to deliver the CRISPR to that tissue that's affected, like the particular organ or cells, and knowing the genetic cause very clearly. So that's something that I get asked about all the time, is like, everyone has a disease or they know someone who has something, and they, of course, it's a very emotional thing, and you want to know if this is going to be able to help you or your family. And for a lot of conditions, it's either too complicated, like too many mutations are kind of working together to cause the condition, or we simply don't know how the genome is related to that disease. So for those, it's going to be a little bit longer, but I think in theory, there's a huge potential for any genetic condition.
So we have time for a couple more questions. Two are more kind of feasibility questions for curing sickle cell using CRISPR. And so I open this up to the whole panel. One is, I guess, Cassandra Marilyn brought up stem cells, how that was, or I think Marilyn, so someone asked, why can't blood stem cells from people without sickle cell be given directly to those with sickle cell? And then another person asked, how do you ensure that the corrected cells are the ones that survive and overtake the existing sickle cell red blood stem cells?
So I can start with the first one in terms of why can't stem cells from a healthy person be given to another person. So that is similar to a bone marrow transplant. So in a bone marrow transplant, a person with sickle cell disease would have to get a donor who matches several different types of criteria. And once they match these criteria, the bone marrow, the stem cells from the healthy, not person without sickle cell disease, will be extracted and transplanted into the healthy person or into the person with sickle cell disease. But that process is a very risky one in some ways, because, and it's also not accessible for everybody, because you need to really find a donor that matches you on a lot of different metrics, or else you can go through something called graft versus host disease, which is where your body attacks the transplanted cells. And, it's very risky, and it can have all of these complications, which unfortunately can include death. So, having something like CRISPR, where you are your own donor, would be a really great step in the right direction for sickle cell disease, because it does reduce that risk of graft versus host. But of course, there are risks, and it is still in clinical trial phase, but that's sort of what's going on when you take stem cells from a person without sickle cell and putting it into someone who does have sickle cell.
Great. And so the last set of questions, which I think will go down the line and everyone has a couple seconds to answer. But since this is part of the Bay Area Science Festival, we have a lot of kids, students, families watching. And one thing that I've heard throughout our conversation is that we need a shift in how scientists think. we need to bring in stakeholders. We need to think about what we're doing, before what we've made is we're trying to give it out to the world. So maybe what do you say to the students listening who want to become scientists? What do you say to them about how maybe they should think about science? Or if they're interested in something they heard today, where can they go to learn more? So let's start with Marilyn, and then we'll go Cassandra.
Okay, so I think that one of the most important key takeaways is that technological solutions like CRISPR really need to go hand in hand with social solutions. And even though, CRISPR and other types of technology can really, tackle the molecular aspect of an illness, without looking at the larger picture, in fact, sometimes technologies can do more harm than good without realizing it. And so I would say, if you're young and you're interested in biotech, find ways that you can supplement your studies or something with thinking about the broader impact on the people who you're trying to treat. How are they going to be able to access this treatment? Is this what the community wants? So I would suggest people start there. But I think it's wonderful that there are so many young people that are, interested in this type of technology in this community.
I'd just like to echo what Marilyn said. If you love science, come on over to hematology. It's a great field. You get to work with a lot of great people. The sickle cell historically hasn't had, just that type of interest that it does now. So, just continue doing what you're doing. If you want to get involved, we're here. Feel free to reach out to us. We can always use all the help, any help that we can get for a lot of our work. And there's a lot of work to be done. So if you do have interest, come find us.
I think it's great that you're tuning into the Bay Area Science Festival. If you live in the Bay Area, there's so many great resources. There's a lot of institutions, and we live in an area where it's a hub for innovation. So it's an exciting place to be if you're interested in science, technology, engineering, and math. What I will echo with what Marilyn and Cassandra said. I think the way I think about my role as a scientist is, science can be a tool for social justice, but we do need to look at the broader implications at the historical context. So I absolutely +1, look beyond your STEM classes and think about how science is everywhere and connects to like every aspect of your life, every aspect of society. And I would particularly say to students of color that your life experiences, your culture, your identities, they're a great asset for science. So, if you decide to go into these fields, there might be times where you hear that you shouldn't bring your whole self into science, but don't listen to that, because who you are is exactly what we need to create a more just society using science.
Megan, do you have anything you want to add?
I don't think I can add anything to that. I mean, I agree with everything everyone just said. You said it perfectly.
Great. Well, thank you so much, panelists. This was a fantastic conversation, and I'd like to hand it over to Megan.
All right, thanks, Kevin. And everyone can turn off their video if they don't want to be seen anymore. I'm just going to close it up. Thank you again so much to the panelists. That was really amazing. I'm so happy to have had you here. Thank you to everyone who tuned in, and especially to you who asked questions. I'm sorry we couldn't get to all of them. Hopefully we can, have another event in the future or something and make sure we answer them all. But I wanted to quickly, give you a little bit of information if you want to follow up on anything. So yeah, this is all of our, contact information if you want to follow what we're doing at the IGI. And yeah, with that, I would like to thank the organizers of the Science Festival as well. So thanks everyone for being here. Have a good night.